The speed of development of COVID vaccine has been unprecedented, basically thumping all expectation. So today we're going to reflect on that process with a legend in infectious diseases, Dr. Carol Baker. The objectives of this podcast is to discuss the vaccine development for COVID-19 and key elements in the initial success, understand the clinical data leading to approval of the vaccines, and comment on the future of vaccines with the implementation of new technologies. Welcome to Editors in Conversation. This episode is brought to you by Antimicrobial Agents and Chemotherapy, a journal available at aac.asm.org. I am Cesar Arias, Editor-in-Chief of Antimicrobial Agents and Chemotherapy, and this podcast is supported by the American Society for Microbiology, which publishes AAC. Don't forget to check our latest issue just published just last week, aac.asm.org, with outstanding papers on resistant pharmacology of antimicrobial agents and clinical therapeutics, in particularly a review of new antifungal drugs that is really insightful and very informative. Joining me today to talk about COVID-19 vaccine is a distinguished professor, as I mentioned to you, a legend in the field, Dr. Carol Baker, who is professor of pediatrics at the University of Texas Health Science Center, McGovern Medical School, and is the recipient of the 2019 Albert Sabin Gold Medal for vaccinology. Welcome, Carol, to the program.
Thank you. My privilege.
So we are in 2021, and I guess 2021 is going to be the year of the vaccine, and where we hope is going to be a better year than it was 2020, what was the year of COVID-19. In your experience, and you know this, and you've been linked to trying to basically eradicate diseases in children and others for infectious diseases and in vaccinology. What are your gut impressions of the development of vaccine against this pathogen and where we are right now with the vaccine?
Well, one word, amazing. It reminds me, because I had occasion in the last year to read President John F. Kennedy's moon speech back in the early 60s, which was a challenge to not people like me, but to engineers and scientists to land on the moon, which we did in July 1969. And I thought that was amazing. It took almost 10 years, but to develop any vaccine, literally, certainly less than a year, but not like one day less. It was an amazing scientific human accomplishment because there were so many things besides science. Of course, science was fundamental, but the collaboration, the cooperation, the consensus that was developed in all the pieces to get vaccine actually for the first time in arms. I've never seen anything like that in my lifetime. I hope going forward, if we have these kinds of challenges, we've always been told in infectious disease, the next pandemic is coming. And I didn't think I'd see one so soon after 2009. So this has been just an amazing time for humankind. And the reason I'm saying this in such a positive way, consensus, collaboration, cooperation. Science is fundamental, but all the other pieces, when troublesome things were going on in our world, I was very impressed. It left me feeling very hopeful. As you said, I'm certain that 2021 won't be perfect, but in terms of the pandemic, it's going to be better.
Thank you for those insights. You've been working in vaccine for Group B strep all your life, Carol, and an amazing amount of efforts to try to reverse this still lethal disease for mothers and young children. When you see these new technologies and this speed that these COVID-19 were developed, so how do you think that could relate with your efforts against bacterial vaccines or pathogens that you have studied so deeply?
So I wanted to be a doctor. That's why I went to medical school, but my background is English literature, 20th century American literature specifically. So I was not trained as a scientist, but when I started my pediatric training, I had these young babies dying. And it's very interesting. That began my work in group B streptococcus, but it also began it really was the beginning of public health interest, because I went into ID in some ways because I thought I could fix things. I could make patients better with antimicrobial agents. But especially if you're a neonate, it's much better to prevent things. So the idea of maternal immunization seemed very natural to me and logical. But that was an idea in the 1970s. In the 1970s, there was no vaccine being given to pregnant women for anything. We probably had some women outside the United States who had been exposed to rabies that got vaccinated with rabies vaccines, but it was an idea before its time. So the science moved along fairly nicely. But you asked the question about new technology. Well, specifically the mRNA vaccines. So those were the fastest ones for very good reasons. But I think that there's technology when you're talking about bacterial vaccines where mRNA is, we may not need that. And it hasn't been explored. It's, as you know, mRNA vaccines were have been used actually for rabies, for Ebola. So there are some parallels. But about, it may be more than a decade ago, Rino Rapalli's group in Siena, Italy, started talking about reverse vaccinology in the context of our ability to whole genome sequence and understand what antigens other than the obvious ones. So for group B strep, pneumococcus, we talk about capsules, and it's worked very nicely. Conjugates are better immunologically, and conjugates, at least for my pathogen, group B strep, work a lot better than pure polysaccharides. But wouldn't it be nice to use other epitopes, target other epitopes? So I think that a routine conjugate, polysaccharide protein conjugate, will work for GBS, maternal immunization, infant disease prevention. However, the big burden of disease in rich countries now is adult invasive disease. And I'm guessing the pathogenesis is different. So my bet, if I was going to develop a vaccine, and those days are over, I had my chance, and the conjugates are being picked up by commercial entities now, thank heavens. I would use one of the surface proteins, and specifically the pilus proteins, because pilus proteins have been associated not only with attachment, which doesn't surprise anybody, but also invasion. And if the invasion in the majority of adult disease is different, if it's, for example, chronic ulcer disease and diabetes, the invasion occurs at the skin site or the soft tissue site, rather than, the typical thing we see in the nasopharynx or the GI tract in bacteremic disease in neonates, then I think reverse vaccinology could find one or more proteins as a conjugate. So you'd have two major targets that might prevent disease. But that's a long answer to a question. And as I say, I think new technologies, the great thing about the epidemic, because there's not too many great things, is that we have been forced to do things as quickly as possible and as safely as possible to control this epidemic. So now for the first time, and I am amazed when I hear people say, pregnant women, yes, pregnant women, from an equity and fairness and ethical point of view, should be allowed to have access to this vaccine. That's not saying pregnant women should get this vaccine, but these words are just amazing to my ears, because my entire career until recently is pregnant women should be protected from getting vaccines, no matter what the rationale and potential safety profile. So it's been very, very interesting. And that one of the good things about the pandemic is pregnant women are no longer, don't touch them, don't give them anything, but consider them equal to other populations. Yes, safety, yes, efficacy, yes, science, but don't put them aside.
Very important points. And that gives me to that question that I had. Do you think this will change the way clinical trials are done to be more inclusive, for example, with pregnant women, because they normally tend to be excluded in these efforts? And you know the rationale for that, and I understand probably, but you know that precludes the study of very important population. Do you think that will be a change in the future on that
I
approach?
think we're already beginning to see the change. Two things happened, Cesar, in the 2009 pandemic, which, as you know, was nothing compared to this one in terms of death and disease and the economy and everything else. The 2009 pandemic took the immunization of pregnant women against flu to protect them, and now we have multiple studies showing it will protect young infants until they can be immunized against influenza. That took the uptake in the United States for flu vaccine in pregnant women from 10 to 15% to 50% in one sweep. And shortly thereafter, because of pertussis epidemics in very young infants, they were dying, they went on ECMO, hospitalization, etc. Well, they were too young to be protected from their 2, 4, and 6 shots, or the 2, 3, and 4 that they use in the UK, for example. So the United States was the first, and I'm very proud of that because I was chair of the ACIP when, by one vote, and you can guess who the vote was, the recommendation to immunize pregnant women in the 3rd trimester against pertussis occurred. Very shortly after that recommendation, of course, the United States has a different healthcare system than the UK. The UK had a horrible outbreak also at the same time. They basically announced that pregnant women should be immunized, and within 6 months, their epidemic in infants was over. The efficacy of the adult booster pertussis vaccine, Tdap, was 90%, and the uptake was way above 80%. And now we have several studies, because this is being done in other countries too, showing us literally in hundreds of thousands of women how safe this is. And that's the thing. You start saying, do I think they'll be included? Yes, I think they'll be included, but we need to have safe trials to make sure that there's something about pregnancy that might be different for a specific vaccine. So on one hand, I'm very pro-maternal immunization, especially given the very poor immune system, even of the term healthy infant, until they're fully immunized. On the other hand, I do think that we need to be careful, and especially there could be things where we wanted to back up immunization into the 1st trimester or early 2nd trimester. Fetal development's pretty much done by early 2nd trimester, but it would depend on what we were trying to do, Cesar, in terms of protection. I think we need to be careful, but I think we need to be careful in all of our populations. Just because you're a healthy adult, we need to be careful and look at safety and efficacy.
So excellent points. So you know this data because you've been in these meetings, you know, when these trials being discussed, and you had to review this data very, very closely with the ACIP and other communities. What struck you as the most relevant information and make you comfortable with the data provided by these at least 2 vaccines that are now available in the United States?
Well, during 2020, as everyone who reads the newspaper, looks at the television, etc., it was a very chaotic time. Despite this, the what we call the work group, the people that looked at the data in depth, and then it goes before the voting members of the ACIP, The dedication and hard work and coming together, the collegiality, I found amazing. One of the things you try to do in a work group is to keep the group fairly small to make it functional. This was the largest work group ever. We had ethicists, we had lawyers, we had every professional society that you can think of that would be relevant for old people, for young people, for pregnant people, the AMA, organizations that push vaccines like the National Foundation for Infectious Disease, and then all the regional and state and territorial public health people, etc. Everybody got input. All the stakeholders got input, which meant we had to do hours and hours and hours of talking and watching the data. The thing that was wonderful about the data, and I don't particularly like this word because it's overused, but there was wonderful transparency by the companies that were developing the vaccines. They were asked questions when their data was presented. And you know, Cesar, if I'm presenting my data, or if you are too, you want to have integrity and honesty and interpreting it, but you may not see something and somebody else may. So sometimes the questions couldn't be answered right then by the 3 or 4 people from the company presenting, but the questions were written down and we got feedback by email or the next presentation, et cetera. The amount of detail, the amount of, we're all in this together. I mean, I hate to sound so positive and dramatic. It was very tiring because all the meetings, all the Zoom meetings were 2 hours, sometimes went 2.5 hours. They sometimes occurred on Saturday and Sunday, which is not exactly my favorite day to be sitting at a computer, doing that anyway. But the detail and the real-time things that were happening, the public-private partnerships was another part that I had never seen in action. So, this was a pandemic, and we had various vaccine manufacturers partnering sometimes with the NIH, sometimes with a small biotech like Moderna is a small company in Boston, Massachusetts. They needed some big time help in bringing the science together with the clinical trials, for example. So, but the detail that things were, the detail, the integrity, the honesty, the questions, people gave them a hard time. And it's because we wanted the data to be the best possible data, to be able to say to the American public, these vaccines work. By the way, for months we were told, because the FDA target for efficacy for the emergency use authorization was 50% and the lower bound was 30%. So I'm getting myself all ready for a vaccine that's 50 or 60% efficacious. And, the good news is the 2 mRNA vaccines were almost identical in their efficacy. And then looking at the safety, so we're being so careful about the safety. We are looking at exaggerated immune responses from vaccination in previously infected people, whether they had symptomatic disease or just asymptomatic infection with COVID-19. The safety measures, so we had several passive surveillance mechanisms set up in the United States, one active surveillance, but it's regional centers. And now the new one, vSafe, which is a real-time iPhone safety mechanism to continue to look at safety in hundreds of thousands of people as we go forward to, again, inform ourselves of the safety. And then the same thing, we just had a call about the effectiveness. So efficacy is in a clinical trial, placebo control. Effectiveness is real data when you're out there immunizing a population. So new systems and the use of old systems have already been designed to look at effectiveness, starting with these first 2 vaccines and going forward. I'm very impressed. And what happened? Why couldn't we do this before? Well, the science is explained by the mRNA technology. What we have cut through in one word is the waiting and the bureaucracy. And I hope for future vaccine development in non-pandemic that we have learned how to be efficient, but continue to be very careful. Science first. Science, science, science. Ethics, which I've always thought was just an integral part of science and medicine. These things need to just be part of our, of our DNA as we go forward in vaccinology. So in many ways, pandemic is horrible, but I think in terms of future vaccine development and implementation, we've learned so much, and we've learned by things that aren't in place and that we have to build. So I'm optimistic going forward. It's going to be a big job.
So following that, I am very interested to hear your opinion about the inclusion in these clinical trials, particularly of minorities in the United States who are being affected. And how when the data that permeated to you and the inclusion of that, were you satisfied with the inclusions of people, brown and black people and minorities, and where was the panel, aware of this situation?
More than aware. In fact, I'm somebody that likes different words in sentences, and I think it's the English background. I would get extreme fatigue hearing the emphasis from individuals about the inclusiveness. But that just tells you how important it was to everyone. And I will tell you, behind the scenes, in the clinical trials, the companies were very much encouraged. You can replace encouraged with whatever words you would like to use, to make sure that they hit target minority groups that were so important, including some that are hard to figure out. So part of the normal ACIP liaison group is the Indian Health Service. And so Native Americans were very much over-affected, and Alaskan Natives. Well, we have special health services through the federal government, but actually being able to reach these people through education so that they would be willing to be vaccinated. And the same with some other minority populations. But up front, the companies developing these vaccines were told, they were beaten over the head along the way, that they must have a certain proportion of minority people in the trial. So they worked very, very hard at the clinical centers. And I know in the Texas Medical Centers, these clinical trials occurred. There was tremendous pressure on the clinical trialists to, yes, the inclusion and exclusion criteria, but really get these people of these important minority groups that are overrepresented by disease, hospitalization, and death in the trials. And they did a tremendous job. And this was not just in the United States. Part of the trials, as you know, were done in populations outside the United States. Diversity is an issue worldwide. It's not just in our country.
Fantastic point. Okay, so we get this vaccine, the data look good, and now we are in the rollout phase. So what do you think about the rollout phase so far, and what is your prediction? Is it going to get better? How are we going to get there?
So let me back up to the ACIP. It was important in making recommendations. We knew we wouldn't have enough vaccine to do the tiers. And a lot of work was done to get data on what the American public thought. And it wasn't surprising to me, but it was surprising to many people that the American public valued healthcare workers because they wanted their healthcare system intact in case they got sick themselves. And the second, and this was a change, so healthcare workers were going to be a recommendation for phase one. There was a discussion about some healthcare workers would be reluctant, but they were going to be first approached. What changed along the way based on data, so this was a science-driven decision too. The public wanted to prevent death and serious hospitalization and maintain our healthcare infrastructure. So added in with the data of how much death and serious disease hospitalization you could prevent, people 75 and older were included. That was the ACIP recommendation for the first allocation, followed by 1B. We knew the whole time that vaccine would go to the states and regions, to the cities, to the county health departments. And we live in the United States. but it is one united country, but it is different states. And so I am not surprised that the states took those recommendations and reflected upon them in a different way. But I will say that the 75 and above added to the healthcare workers in Tier 1A was based on data of how much disease you could prevent, how much death you could prevent through a lot of different modeling and a lot of hard work. Of course, everybody wanted to go down to 65 and to the people who had comorbid conditions, one or more. Many of the very sickest people, as you know, as a clinician, have 2, 3 comorbid conditions, making them susceptible. So, and we continue to do that. And while everybody's saying, yes, we'll get to children, yes, we'll get to pregnant women, there was a lot of pressure to make the companies, again, I'm talking about the mRNA companies, to start thinking about beginning trials. And I will tell you that both Moderna and Pfizer are doing trials, have begun trials in children. So the original Pfizer, the very first one, was down to 16. They're both going down to 12. And I think this will be a very slow progression down to young children. Do I want to predict when young children? So if you want to talk to me about when do I think an 8-year-old will be immunized, not this year, because what they're doing with children is different than what we're doing with adults. In adults, we have phase 3 placebo-controlled clinical trials for the EUA, accumulate data for licensure, like our usual vaccines. What they will be using for children is what we call bridging studies, and that will be based on serology. That will be based on us finding, through vaccine failures in adults, that's how they'll figure this out, what the protective level of neutralizing antibody is, because we don't know that right now. We've used a correlate of protection of convalescent plasma exceeding that level of people who recover from the disease. And the vaccines seem to be as good as or better than convalescent serum from infected adults. So it's going to take a long time. 2022, and a recommendation for pregnant women, unless you're talking about third trimester, where, you know, the baby's just sort of feeding and growing in there. I don't think we're going to see that anytime soon. What I want to see is the populations for which the vaccine has been authorized. I want to see good allocation. There's been bumps in the road. I want to see good stewardship. I'll use that. There are shortages, and they're going to be. We need to give the most vulnerable people vaccine first. And the fairness of all this, hey, I live in the real world, you live in the real world. There are some people that are getting vaccinated that, you know, are getting vaccinated because they know somebody who knows somebody. But we've really tried to be fair and equitable and target those most vulnerable.
So following on that, there is a lot of talk now about the doses, and that the vaccine for second doses may not be available, and even recommendations for our local health system indicates that, it's okay not to give the second dose because maybe you are okay. So what do you think about that?
Well, let me start with science first, because we have to be science first. You know this, the published trials, the data that's available for the public to look at if they want to, had for Moderna vaccine, the second one, an interval of 4 weeks, and for Pfizer, an interval of 3 weeks. So I like to be pure and say, we know this works. And we're going to see some more data coming in those original trials that will tell us what proportion of people might be protected after one dose, at what time. Okay. However, biologically, I do not see a reason from a biologic point of view why delaying a dose, and in fact, there's clinical guidance now from the CDC that I believe says you can receive your dose as late as 42 days, your second dose, after the first dose. We'll have more and more data that will help guide this. And you're aware that the UK, when they had their one vaccine, almost immediately decided that they were going to give as many first doses as possible. And when they got second doses, fine. Again, biologically, that's plausible that you don't need to hurry, but you are delaying the 95%, 94% effectiveness, efficacy that's been reported by that interval. So I don't know. I sort of don't like people making up things, but I'm a clinician, and my whole career I've had to sort of make up things depending on what was available and the individual patient. But I think people should be very thoughtful and very careful about what they're doing. They need to be justified in saying, well, we're going to give everybody first doses. Well, everybody must include vulnerable minorities and vulnerable populations that should be first in line. Or it's just not right.
And as a corollary of this, now we are facing this emergence of these variants, which we don't know much about. We know they are more transmissible. We believe that potentially will respond to vaccines, but that means the rollout of these vaccines probably need to be more urgent. And that may push these situations that you just mentioned of just trying to maybe sort of forfeit the second dose for just trying to vaccinate amongst people. So what do you think the influence of these variants are going to have in this vaccine rollout?
Well, the whole story of COVID-19 is I was forced to give opinions with insufficient data. So I'll answer your question in that context. These variants, coronaviruses, I am not a virologist. I don't like viruses. You can't see them in a plain microscope, okay? But I've learned a lot about coronaviruses. Their very nature is that they rapidly mutate. And not necessarily the SARS, but they're going to keep changing, and there will be variants. One of the good things about this more transmissible variant is that it has spurred funding to look very hard at variants and look at what does that mean. We know there's variants have emerged and they haven't meant anything up till now. So that's going to be looked at much more carefully. You may remember it was only in 3 states. And I'm like, well, that's because those 3 states had systems set up ahead of time to do the work to look for variants. It's not a PCR test in a clinical setting. I think we will see more variants right now. There are data suggesting that the variant is in the spike protein. It makes it more transmissible, but the vaccine antibodies being elicited so far look like it should work just fine. But you're absolutely right. The urgency to get our population immunized as rapidly as possible, I think we will begin to see more expert opinion, clinical guidance. I think that we are going to see some delay in second doses. We're going to have some delay because human beings are human beings. They may not show up for their second doses. That's been a concern the whole time. And we have at least one vaccine coming fairly shortly, I would say within weeks, a product of Janssen that is supposed to have a single dose efficacy trial. That would be wonderful. Again, we need to make sure that it has good efficacy and very good safety. It sounds promising from the announcement. The other thing that's been interesting, you know this as a published physician scientist, you're not supposed to tell your data until you sort of have your paper peer reviewed. Well, so much for that in the era of COVID-19. So now we have press releases that tell us what's happening scientifically. But it would be wonderful if we had one or more vaccines that would work with one dose. And I think we're going to have some flexibility on the dosing schedule.
Is there any push from ACIP or other organizations for the companies to do trials on these variants, or are you aware the companies planning any trials on the variants?
The companies are very aware of it. ACIP is not pushing, but other organizations are pushing, again, to have better surveillance. It costs money to look for these variants. It's very expensive. And hopefully some of the new money for COVID-19 control is going to make our surveillance much more robust. But as you say, the fact that there's a variant, there are going to be variants all the time. We need to know what the meaning of the variant is. And so this one that we know about is much more transmissible. And it's one of the reasons we're having a surge. It's not the only reason. And the last thing to say, I find it very disappointing, but being an English lit major, I've read a lot of stories about humans all over the world, centuries ago and in modern times. And I think that if we don't follow mitigation strategies while we're trying to get there with immunology and vaccines, we're just doing other people a disservice and ourselves a disservice.
Couldn't agree more with that, Carol. So we have a new CDC director, an infectious disease physician. If you were to advise Dr. Walensky right now on the future pandemics and looking back on what happened in the vaccine development, where do you think we should emphasize to prevent another catastrophe like that happened last year and still this year?
Well, human beings are human beings. The reason we have a fire department is if your house is burning down and the firemen come and put the fire out, that's very visible, etc. And we don't mind that the firemen get to sit in their firehouse and cook meals and get some shut-eye, read some books, whatever in between. Fire is very important. The problem with vaccines and vaccine preventable diseases is that we tend to be reactive. We don't do anything about a problem till it's a problem. So after during the 2009 pandemic, I was on the ACIP and there was a big push to, we weren't prepared. Okay, we weren't prepared. The public health people had been talking about not if, but when, and we weren't prepared because people don't prepare for something that might happen. Oh, we don't know when. Oh, it probably won't be in my lifetime, etc. So it's not the public health people that don't want to plan. I think we will learn a lot from this pandemic. And whatever we learn, we need to keep in place. We need to invest money in doing things and be ready ahead of time. So vaccine technology has helped us so much in this pandemic, but the whole infrastructure, the whole allocation, the way we deliver vaccine, we're going to learn so much, and we need to leave many of these things in place. The one thing I'm not hopeful, because this was discussed so much, we have learned, well, I didn't learn one thing. When I started medical school, starting seeing patients quite a while ago, there were incredible inequities in healthcare and in minorities. And the minorities have changed because some of our immigration patterns have changed. We have more of some people and less of other people, but there were health inequities. And one of the things that was discussed over and over and over is we need to address these inequities. And we do need to address these inequities. But the reason I got frustrated is we have a pandemic. Our job is to get a vaccine out there. And we're not going to be able to improve our healthcare system just in addition to everything else we need to do. But we will learn so much about our ability to reach racial and ethnic minorities. Use this knowledge. You can't communicate to all populations the same way. And that's a really important thing. I learned in pediatrics. I came from a very my growing up white middle-class background. Okay. I learned how to talk, now that people use this word, I didn't know, in an ethnic and culturally competent way, appropriate for their experience. I was horrified that there were children who'd never slept in a bed. There were no beds in their houses. I saw rat bite fever because these homes had holes in them, and there were, rodents that came inside. That was part of their normal life. So I hope we can begin to build on the information about how to communicate to populations in a way that we will rebuild trust. Because fundamentally, with something's coming from the federal government, whether you're talking to a state governor or a state health department person, they have to trust that you're telling them the truth about the science. We have to convince people that these vaccines will work. They may not feel good. After I had my first dose, I had a headache, and I just never have headaches. It's not my target organ of stress. And I went around all day. Now I could have taken, something for my headache. It took me the next day to realize that it was a minor side effect of being vaccinated. So clearly it was not an important one. But we need to be truthful. We need to not over-exaggerate, but we don't need to underplay that there are some side effects in a small number of people that may make you feel bad enough to not be able to go to work for a day and possibly 2 days. That's the kind of, my word I hate, transparency that we need to rebuild trust.
Dr. Baker, thank you very much for this wonderful conversation and your wise advice and perspective. This is Cesar Arias, Editor-in-Chief of AAC, signing off. Thank you very much.